Again, sorry for the confusion. Thanks for recording, Monica. All right, welcome everybody to, oh, the date is wrong, March 11th. I have the wrong slides up, that's why. Apologies for that. June 8th, there we go. Looks like the right date. Alrighty, so we've got a pretty packed agenda for today. We're going to be hearing from some guest speakers, Bill Robberson and Dr. Rob Andrews from University of Toronto. And then at noon, we'll transfer over to hearing from our work groups, Brent Alspach from the Consumer Messaging Work Group and Shelly Walther from the Sampling and Analytical Methods Playbook Work Group. We've got a few other updates after that, and then some time for discussion, and then we'll recap and adjourn the meeting. So just to bring everyone up to speed, as you all know, we've got two legislative mandates that require the State Water Board and the Ocean Protection Council to address microplastics. Senate Bill 1422, just to give everyone a brief update on where we are with that. We are planning to release the response to comments and the updated policy handbook by the end of this week. If that's something that you're interested in, I'll be sure to update the subcommittee listserv with that information. Looking to have a board meeting late July or early August. We also will be providing ELAP accreditation by the end of this summer. In terms of the aquatic ecosystem, Senate Bill 1263, we adopted the statewide microplastic strategy earlier this year, and we're moving forward with some additional items, working closely with the Ocean Protection Council on these mandates. We'll hear a little bit more about this at the next meeting from Caitlin Kalua.
Some of them writers may want to come out and take a tour if they have a question about the facilities. We don't see that so much at IEUA as opposed to what they see at the RCA.
Not sure if that was relevant to this meeting, but maybe someone was talking with their mic open. So just going to mute. Alrighty, so sorry about that. We have some completed research that is going to be discussed further in these work groups. We adopted the definition of microplastics in 2020, and that definition has formed the basis of the work for the method standardization. It's been completed and is now ongoing for additional matrices outside of drinking water. Also developed some human and ecological risk assessment frameworks and a living a database called Tomex that is now live. And that brings me to the subcommittee and our goals. So again, for those of you that are just joining us, this is a local and global community exchange of information and data for microplastics monitoring methods and tools, and it's open to anyone that is interested. My co-chairs include Dr. Monica Arienzo at the Desert Research Institute and Shelly Moore at the Moore Plastic Pollution Research Institute and San Francisco Estuary Institute. The organization of this work group is nested in a number of different hierarchical work groups. So this is part of the Water Quality Monitoring Council. The Trash Monitoring Work Group is a subgroup there, and this is the Microplastics Subcommittee of that work group. There's 3 other subcommittees of the Trash Monitoring Work Group: encampments, citizen science, and data standardization, management, and coordination. And this microplastics subcommittee, currently we have 2 working groups, the consumer messaging and the sampling analysis playbook work groups. I'm going to skip over this. This is a bit of a recap of why we're doing the work that we're doing right now on drinking water, as well as a little bit of the representation. I covered this sort of in the last meeting. What we're really trying to achieve here is stakeholder-driven priorities. And so one of the previous meetings, we collected a poll about what our stakeholders are most interested in working on, and that includes sample collection, analytical methods, and communication strategies, hence the 2 work groups. Oh, my slides are not showing. Okay.
I see them. Is anyone else not seeing them? I can.
Oh,
okay.
I can see them.
I can see them.
Alrighty. So the focus on sample collection as one of the highest priorities for our stakeholders brings us to the two presentations that we're going to have today from Bill Robberson and Dr. Bob Andrews. That information will feed into the sampling and analysis playbook that we're currently developing. This is to assist anyone working on microplastics, whether it be in industry or academia or research or government, what have you, to have harmonized methods that are reproducible and of high quality. So for drinking water, ocean water, wastewater, sediment, and fish tissue, we have analytical methods that we're developing both for the sampling and the analysis for Raman and infrared spectroscopy. So this playbook will provide additional guidance on how to use those methods. And we're building from some prior precedent and knowledge here. The California Trash Monitoring Methods and Assessments Playbook was published a few years ago as part of, again, of that trash monitoring work group. And we have the leads from that project on this microplastics subcommittee work group. And really, we have a pretty ambitious goal with the work that we're doing through this subcommittee. It's not just to implement California's goals, but also to lay a foundation for a national monitoring network, which was a recommendation from the National Academies of Science, Engineering, and Medicine in late 2021. We'll be talking a little bit more about the sampling protocols today, but just to remind you what's out there, the ASTM committee released a sampling protocol for microplastics and various water matrices, and we currently have a contract with Brown and Caldwell, SQUIRP, and the University of Toronto to optimize and evaluate these sampling methods, comparing alongside the ASTM method, a novel inline filtration method that you're going to hear a lot more about today from our lead on that, Dr. Bob Andrews. And again, we've got this is a standardization project that is currently ongoing with SQUIRP, Brown and Caldwell, University of Toronto, paid for by the State Water Board. And the goal is to have this monitoring method, or sorry, sampling method online and evaluated by next year. And this work group will act as a iterative stakeholder engagement to carry this project into fruition and make sure that the insights from this are integrated into the community. I mentioned before, we have this playbook work group, and we've got the project leads here, Shelly Walther from Los Angeles County Sanitation District, and Shelly Moore, the co-chair from the Moore Institute. And the goals are to ensure access to information, tools, and resources, and a high degree of quality control for sampling and analysis of microplastics, harmonizing reporting. And currently the goal is to have a playbook developed, ready by spring of 2023. And this includes some videos, kits, resources, in addition to a document that can be used. And just to give everyone a heads up, we released a revised version of the infrared and Raman spectroscopy methods for microplastics in drinking water just May 27th, so about 2 weeks ago. That's on the State Water Board website. And so this is the first revision of this method. So make sure if you're using that to go and download that new method. It's on our State Water Board website. We also will be requiring some water systems to report microplastics concentrations in source waters and drinking water in their consumer confidence reports. This is part of the Senate Bill 1422 requirement. And what we know from the literature is that the general public is very concerned about microplastic pollution. When it comes to the marine threats, they rate plastic pollution as the #1 threat, above chemical pollution, loss of marine species, collapse of fish stocks, some other major threats. And this holds not just in the United States, but across 15 different countries. And so we have the public is primed to be concerned about this issue. And so we need to be able to respond to them with adequate information, reliable information, and be aware of the predisposition that the public has. There's a lot of information out there about the human health effects and the ecotoxicological health effects. The World Health Organization released a statement in 2019 effectively claiming that there's no evidence to indicate a human health concern, which is not to say that there's no evidence to indicate that there isn't a human health concern. But this type of messaging, this uncertainty messaging, it makes it really challenging to to convey risks or potential what this actually means for people's health. And this soundbite is often or has been interpreted to mean no risk, even though it really is no evidence of risk. We're leveraging some concurrent efforts here with a Water Research Foundation grant, Project 5155, with Brent Alspach as the principal investigator. Brent is also the chair of the Consumer Messaging Workgroup. You'll hear an update from him in a bit. And the goals for that work group are to effectively and transparently communicate the risks of microplastics to consumers, while also increasing the trust in public water agencies. The deliverables for this are very similar to the other work group. Spring 2023 is our anticipated deadline for some suggested consumer confidence report language. And then there's a risk communication toolkit that this work group is developing that's will be an ongoing effort. Another quick update, microplastics data sets are incredibly complex, and there's a lot of information that can be held in one sample that can be useful for a lot of different items. We don't currently have a database in the State Water Board or elsewhere that can intake this data in a harmonized and useful way that doesn't compromise the complexity of the database. And so we're working with Dr. Wen Cowger, the People Lab. We've got Rohit and Ryan here, as well as the State Water Board, to develop a harmonized data reporting pipeline, at San Francisco Estuary Institute as well. This is a draft flowchart for how this data would be taken into the State Water Board and integrated into our existing data systems, as well as some open source repositories and tools that live outside of the State Water Board, like OpenSpecKey, Plastiverse, and Tomex. And so we'll hear a little bit about this from Wen near the end of the meeting. And again, we've got some project partners listed here for that project. And we also have a Google Drive repository that is open to anyone, bit.ly/microplastics committee. There's right now, it contains links and information to previous meetings. We also have some publications relevant to the committees on there, so go check that out. We're also trying to get a Zotero library set up so we can share references, and any code that we'll be working on will be hosted on a GitHub repository that'll be shared. So back to the agenda for today. Went a little bit over on the introductions, but I think we'll be fine on time. I'd like to introduce our first speaker, Bill Robberson. And Bill, if you're here, I can give you co-host abilities so you can share your screen and get us started.
Thank you, Scott. Let me see here. Trying to move around this thing here. Get the view bigger. So let's see. I'm going to keep this fairly quick. Okay. All right. You see that initial slide?
Looks great.
Great. So, no, thanks, Scott. So, I don't know how many people are familiar with ASTM. I can't see a show of hands, but I'll assume that people have some familiarity with it. And we started working on microplastics now, well, maybe 4 years ago, 5 years ago, I forget. And in doing so, we decided to start at the toughest place, would be to collect samples from wastewater. And I have on this slide a picture of many of the participants. So for those of you, again, not familiar with ASTM, it's an international organization, American Society for Testing Materials. And the entities who played a major role in the microplastic side have been the California Department of Public Health. Their people are the Richmond Lab, Pima County Water Resources, where we did actually most of our wastewater sampling and preparation work, along with the University of Arizona. The ASTM D19 committee has, I'm going to guess now, maybe 70 members I take it, ASTM has hundreds, ASTM D19 has hundreds. The D1906, where we're doing this work, has about 70 members or so, I think. Adjourn's been very active with us. Frontier Labs out of Japan, Tokyo University as well, Perkinelmo and Shimazu. So shout out to all of them. So we approached microplastics, as I mentioned, from a wastewater perspective, because we figure if you can find microplastics in a sample from raw sewage coming in at the influent stage, then you're doing pretty well. And the problem with collection is when you gather a sample in something like wastewater, you want to not contaminate it. You want to build some standardization into the process. You clearly don't want to use plastics in that part. And you have to develop some parameters upon which a sample can be gathered and prepared. So we looked at wastewater and we said, and drinking water and even ambient water and I'll say open water, such as the ocean or an inland lake, and gathering samples in situ, and put together a parameter of turbidity, or, for those of you who are in the water business, you understand turbidity, that if you've got something under pressure, what's pressurizing it, how that might be contaminating it, be it the service lines or pumps. And if it's not under pressure, how do you gather it that way? And so we built a system for open water sampling. We had one built and we built our own. And with regard to wastewater and the challenges in the fact that not all wastewater systems are the same, treatment plants. You have various ways of bringing your influent in and pulling out the initial grit and that kind of thing. So we built it to be able to take it under hydraulic pressure, hopefully just a gradient. And we also built it to be able to take it with dropping stainless steel sump pumps into certain little tanks. So once you collect it, and I'm not going to show a bunch of pictures of it, but you can imagine that when you're putting together a standard, it has to be vendor or equipment agnostic. It needs to be able to operate in all sorts of conditions. And let's just say that you need to be able to get a result that can be brought into a laboratory to prepare it and then used for various types of analysis, whether it be spectroscopy, Raman, and IR, or even pyrolysis GCMS. And we actually were working on a third characterization process, which I'll introduce in just a minute. And also talk a little bit about the progress we've made with FTIR and LVIR and Raman, and a bit with the pyrolysis side. In fact, I'll take questions along the way, or do you want me to wait, Scott?
Oh yeah, it's up to you.
Well, we can wait. All right. So challenges, you can imagine, reproducibility. QA, QC, I worked at EPA for many, many years. And if there's anyone religious about quality assurance and quality control, it's the U.S. Environmental Protection Agency. And the reason for that is practically everything that they do has to stand up in a court of law. So quality assurance, of course, is about writing that sampling plan. What are your sampling objectives? What are your data quality objectives? Quality control being, how do you manage that quality in the field and in the lab? And so we took it through our rigorous QA, QC process in terms of building a standard. And that's built into the ASTM standard right now. The QA part of someone's sample plan and those data quality objectives, or even just the sampling objectives, they can be all over the map. And I bring this up because you can't really perform, at least at EPA you can't, and I don't think anyone wants to spend money needlessly. You really cannot do a sample and a prep and any analysis without having your objectives very clear. So that really starts with your sampling plan, because your sampling plan is going to impact, potentially even contaminate your sample, and whether or not you get the right equipment. And I mean, in other words, are you looking for just a snapshot in time, with regard to, say, let's take a water column sample in San Francisco Bay. If you're doing just a snapshot somewhere, you know, how much you're going to filter. And if you're going to filter X amount, we set a standard of about 1,500 liters, I think, in open water, because in a lot of our early work, we realized that you needed to have a specific volume to be able to carry apples to apples. So you build that sampling plan with those objectives in mind, and those will actually drive the sample collection equipment that you use, the amount that you sample, where you sample it, and that sort of thing. So that at the end of it, when you've got this sample that's about to be prepped or even has been prepped, you can articulate what it really means with regard to, you know, oh, is the world contaminated with plastics, or are we just looking at non-plastics here? So the collection reproducibility is such that we were able to reproduce it quite often in a couple different plants in Arizona. I know that in working with Casa and Shelly, Walther, and others, and Charles, I suspect you're on right now too, that they've had some trouble replicating the successes that we had. And again, I say that it has a lot to do with the equipment you're using and how the plant's designed and where you're trying to grab it, and your data quality objectives again. So I point that out there, that there are challenges in doing things in wastewater. I think when you switch to an ambient water, be it a bay or a harbor or even freshwater, you would think those challenges would be less, and they generally are. But once you get a sample and you prepare it, you have to remember that, are you looking for a size and a shape? Are you looking for surface area? Are you looking for mass? Those are very important questions, and ones you need to answer before you even go out and collect. So once you've collected your sample, and you know, it's a pretty simple process, really. You know, you filter it, and Scott, I'm looking forward to seeing your advanced filtration process. We've tried many different types of filters, stainless steel, various polyethylenes, that sort of thing. And remembering too that it can be okay to filter samples with a polymer fiber type filter media. You just have to bias out that as part of the results of what you're looking for. On the prep side, well, let's just say that we spent a lot of time and a lot of blood, sweat, and tears on this on the wastewater side, and finally got it down to about 1 day. Our initial process was more like a week, and of course that was not going to fly in a lot of places. Now, Charles, I know you've had some problems, you said, in digesting some of your wastewater samples. I still think that we ought to get together with you and sit down and walk through it. Suffice it to say that if you're going to be using spectroscopy, you've got to get rid of, especially on the wastewater side, all that organic matter, or as much of it as you can. And it just takes a little bit of patience sometimes. So the prep that we did, we then brought forward and used it in both LDIR, laser directed IR that Adjourn makes, FTIR, which is, many people have FTR units, and Raman. And I'll talk about some of the results from that in a minute. We also, since then, have brought it into pyrolysis GCMS. Got some really good results there. And I'll talk a bit about that in a minute too. We did build a sample reference standard for calibrating and for proficiency in the lab. And we built it for both mass and for counting, for count. And anyway, I'll get into where those standards are in a minute. The data quality objectives and data interpretation, again, can't stress that enough, that if one has never walked through the process of building your data quality objectives, EPA has a very good manual for how to do that. And if not, I think that Harry Allen, he's in Guam right now, but Harry or I could teach a course on that. We do it just for free if someone wanted to see it, because without that, I think things are going to be, data's not going to be very good. So the status update, on the ballot, or at least on the ASTM standard side, we do have a collection practice and a preparation practice. Our standards today, they're practices, not methods, because they're not an analysis method. They're practices. And Charles, again, we'd like to hear from you and Jared and others. I think you know all this already, that any lessons learned, because we do think that things, once they're used in the field, a lot more can definitely be improved. Be very interested to see, what you've learned, and to maybe bring that in and go through another round. The sample development practice, that went through ballot. It had no negatives. There was a work group just a couple weeks ago in Atlanta that Todd Gwynn led. Before we bring that through to a final practice, we wanted to get together to see what some things they've learned and see if we should be incorporated some of that. The pyrolysis GCMS, someone just waved down here. Are you trying to get my attention? It's not. All right. On the pyrolysis GCMS side, that practice has been really great. We have built the process for 12 polymers. We do have a calibration standard that needs to be constructed in order to calibrate your machine on a daily basis, or say every 20 to 40 samples. And of those 12 polymers, it helps to have some really good software. Frontier Labs out of Japan were the ones who did all the early lab work, calibration, things like that. We now have 5-point curves for our work. We've got 3-point curves, and we've done some really extensive work to be able to ensure that a laboratory can be accredited through some of the standard accreditation programs. On IR and Raman, we had high hopes for Raman early on, and we still do. Challenges with Raman, I think you may know, they can go fairly low, but it's almost a very, very lengthy process to be able to hunt. We started working early on with some Raman vendors who are using multiple array and various types of settings. And we think it's really great if you've got a very small sample, just trying to confirm some particles. But we haven't taken it through in a larger range yet. So Raman is sort of parked. IR, on the other hand, we, of course, because Agilent has been so active in helping us, they actually gave us one of their new number 3 off the line LDIR machine that we took down to the University of Arizona. And and any new equipment, can have issues. It worked really, really well until there was a power failure and we burned it up accidentally. And then, as you know, COVID hit, and COVID really threw us all for a loop, because lab work stopped in its tracks pretty much early on. And so about halfway through COVID, if I look at COVID as a 2-year process, about halfway through COVID, we decided that, okay, labs are starting to get back together in a small way. And we did a control where the California Department of Health Services, out of the Richmond lab again, Jeff Wagner and his team built our reference samples per our reference sample draft practice. We had the University of Arizona in Pima County, thanks to Jeff Pravat. Jeff Pravat, by the way, shout out to Jeff, was amazing in putting a lot of this together. They took the samples, they prepped them, sent the samples to the Richmond lab, the California Public Health Richmond lab. And that's where Jeff and his team prepped them. And we provided both what we consider to be a drinking water sample, which was essentially MQ water with spiked, and a prepped wastewater sample spiked. So the only people who knew what was in them were the Richmond lab, and sent them off to some very respectable manufacturing laboratory, OEMs, Agilent, Shimizu, and another one. And I will share with you that you know, when you turn this kind of stuff over and a proposed method and say, look, you know, just document everything you're doing, follow the draft method. But if you think, you know, do it one way, do it another. If you need another sample, we'll give you another sample, do it all over again. And we had the best and the brightest. And honestly, I don't think we did better than about 60% recovery, even of the, even in the drinking water sample, with what we're looking for. I'm not going to go into the issues with the slides and things like that, because, you know, we had people, again, some of the best in their field, trying different ways of extracting and that kind of thing. And the results are being published right now, and you'll see them soon if you haven't already seen them. And a lot of good lessons to be learned. So we paused our method development there. And Scott, I think I proposed to you at one point that we sit down with you and the work that you've published and see if we can put these lessons learned together and maybe run through an ASTM ballot with that product to well, actually also run it through in some laboratories too and see what we can come up with. So that's my proposal to this group. We do a dynamic imaging size process that we balloted just recently. It's got some negatives, but you know what doesn't. And when you first go through it, it's got a lot of promise. It's clearly, you know, really looking for characteristics as opposed to some broad detection, I would say. But that's in post ballot as well right now, and we're hoping to move these things forward in the very, very near future. And let's see. And then I threw these in here just to remind people of some of the characteristics we're looking at with regard to count, the ability to identify those targets. Again, isolate individual particles, which, you know, if you've got a large wastewater sample, you're not going to be able to shoot all that. It's going to take you, and even some of the best IR machines, going to take you, you know, a long time, all day, to do most of a slide, even with your fabulous software, gridding out that slide. So, you know, you'll be looking at statistics here. And remember about statistics too, is that as Harry Allen, his coffee cup says, statistics means that you just don't have to be certain about what's going on. So positive identification, very important with regard to the polymers that you are looking at. And then again, you know, a lot of people do want to know size and count. Personally at EPA, we actually evolved to the point where we felt surface area was far more important, because surface area is where your POPs, your persistent organic, are going to accumulate. And if you want to start looking at, you know, potential risk, or I should say potential exposure, excuse me, you're going to want to know that surface area. And you can do that from particle size and count, but I think you're really going to have to extrapolate that surface area out of there too, if you're looking in that direction for issues. And then mass. You know, I don't think that there's a better machine for forensics in organics than PyGCMS, because you're taking the best of GCMS, you're taking the, you know, the ability, for sure, destroying your sample. But with the right target ions and the right reference ions, and the ability to extract that mass in a really wonderful way, boy, what a world PyGCMS has opened up for us in the plastic side. And of course, you get mass. And then just throwing this out here too, I thought I'd give you a little more, because there are probably many of you who are very interested in this. That exercise we did, I take it back. The standard itself is meant to be single lab, that at ASTM, and that gives you about 5 years to be able to go through and do a very broad multi-lab standard. So what we did with this recent exercise was not an interlab study. It was really just a repeatability exercise with those 5 labs. And I mentioned before that we did both a drinking water and a wastewater version. We only used 3 polymer types, polyethylene, polystyrene, and PET. And I think I mentioned what we did with those samples, just want to have a sense of that. And that's what I've got, Scott. Any questions? Any questions?
We do have a question in the chat, and after that, if anyone wants to just go ahead and unmute and ask, that'd be great. So Suzanne Brander was asking if there's any overlap between the microplastic standards that ASTM is working on and the kit that was created last year by the Center for Marine Debris Research, led by Jennifer Lynch.
You know, we worked with Jennifer early on. We met her in Hawaii and looked at a lot of her work, and really so happy that NIST exists. There is no overlap, and there should be. Now, I think Jennifer showed up at that reference sample meeting in Atlanta a couple weeks ago that Todd was running, and I'd like to hear from them. I haven't heard back yet on the process of that, but there should be. And it's one of the reasons why we kind of held off, because we think there needs to be a little bit more participation on that, at least for it to be a standard. So we have some pretty good, we feel we have some pretty good foundation for it, but it seems to make sense to do that overlap first. Answer your question, Scott.
Thanks, Bill. If anyone else has any questions, you can either write it in the chat or just feel free to unmute and ask. Violet Rennick from OCCN asks, can you talk about the pros and cons of the ASTM sample processing method versus other known methods, such as wet peroxide oxidation for wastewater samples? Is WPO an option for sample processing, given some of the challenges experienced by SQUIRP and CASA?
We don't believe it is an option. It's too harsh. And then it depends on your objectives too. If you're looking for polyethylene or polypropylene, maybe not, right? So it depends what your objectives are. Again, we felt that in order to be and the word honest isn't it, but the most authentic with regard to a prep, that you ought to try to not destroy anything if you can. I mean, we even got to the point where we started out, with your magnetic stirrer, right, in flasks, and found out when we were putting our reference samples in there, we're ending up with more microplastics than we started with. So, there are a lot of lessons learned in the prep that we put together.
Wynn, go ahead.
Thanks. Yeah, this is really great. I'm always excited to hear what y'all are up to. I'm interested in the ASTM method and how it's being delivered to the public. So from my understanding right now, you have to pay to get the document. Is that correct?
Oh, Wynn, that's a great question. Yeah, you have to pay. ASTM, it's funny, I've thought about that too, because 2 points. One, the ASTM development process is volunteer. And whether you're an OEM of machinery and instruments, or you're an active lab, you're devoting that kind of time is not cheap. And then people like myself and Harry and Shelly and others, time goes into it. And then they sell it, and you've got to pay for it. On the other hand, they provide such incredible support, both in the collaboration sites and the meetings and everything else. I'm surprised they don't charge more. So the answer to that is, yeah, you have to pay. They ask us when we're developing things to not share those drafts outside of the sub work groups and the work groups and that kind of thing. And that's to keep some integrity to it. I will tell you one quick story. We did have someone who hadn't been participating jump in at the very last minute on one of our developments, just as the ballot was about to close, had never seen anything, never participated, and threw a bunch of Molotov cocktails at it, none of which stuck. But that is not how ASTM works. ASTM, you put broad word out, you get as many people as possible, try to get the best and the brightest participating. And it really is an incredible process. I want to compare that to EPA's method development, however, because I was at EPA for 32 years and dating myself clearly. And we initially wanted to do an EPA standard. And of course, one of our, Anna Marie Cook, who was the main person who, drove this whole process along, she worked for ORD, as she out of Region 9. And we thought, and with Anna Marie in the room, go, yeah, we should do an EPA method. We all dove into it, and she looks at us and goes, don't even think about it. We won't be here that long. So and ASTM really brings industry together outside of that sort of government umbrella to be able to get things done. And it really is an efficient group. I can't say enough good things about the people who volunteer their time to make this.
Yeah. I wonder if there's some way in the future we could make these documents open access. If we could kind of lobby the ASTM to make these documents more available for people, and even have licenses that are permissible to adapt. So that way in the future, like when there's a new method, or kind of like somebody wants to tune something, they can say, well, I don't need to do the whole study all over again. They've already written up the report. The method is here. I just need to change this line. It should be a larger size of cylinder or whatever, and that improves the method by this much. So I don't know, just something I've been thinking a lot about with the ASTM group in particular, and some of the methods.
Yeah, well, just as an invite, any ASTM work group is open to people started participating. You don't have to necessarily be a member, I don't think, to start participating. And we love to have that kind of input. And also too, I don't know how many standards people have looked at here, but if you look at the appendices for our PyGCMS proposed method, it's longer than the standard. And we literally, no, the important part is that we walk through everything that we considered, pretty much how we did it, why we did it, so that people don't have to make those mistakes again.
Right. Yeah.
I'd love to have someone like this, because it really is. It's a collaborative effort, and it'd be a great thing to have that kind of thing. Have someone like you participate.
Yeah, appreciate it. I'd love to in the future. I think in the past, I was informed that it costs money to participate as well. So then I wasn't able to do it, but yeah, I'll keep my eyes out for those kind of opportunities. Thank you.
Well, $75 a year for your membership, and you're in.
Oh, okay.
And it's $75, it's $75. But but your membership every year gets you a free, whether you want digital access, or I usually get the hard copy. So over the years, I've been accumulating, some get old, but standards books, right? And you can even go into space exploration standards if you want, and get any book you want. So for $75, you're getting a pretty good deal. And to share that with you, I don't work for ASTM, but I'm making a big plug, clearly.
Yeah, thanks.
Sure. Anything else, Scott?
Got a question from Shelly, and then I think we should hand it over to our next speaker.
I'll defer to the next speaker. I put some things in the chat. Thanks.
No, let's hear it. Actually, Shelly, you could probably answer them, so yourself. But
yeah, I got involved with ASTM. Bill and Harry and Anna Marie invited me several years ago, and it's been a really nice experience to have so many top labs in the world coming together to do this work and to really look at it critically. Has been really nice. So I am concerned about some of the other labs that have been trying to troubleshoot for a long time. I think there needs to be more collaboration for sure, so that we can make sure that these things are workable, so we have something standardized and feasible and repeatable. So that's what I have really, that's the focus of these ASTM methods, exactly. So I put when you're asking about kind of open source methods, and though the methods themselves may not be, they may be behind a $70 a year paywall, you can see them published in other sources. And in fact, I just put a link to something that's available. But yeah, I highly recommend that folks that are having trouble, that's the nice thing about ASTM, they can kind of really help troubleshoot and make sure that the methods are being followed.
Well, Shelly, thank you for that. I just want to say it's not just a $75 paywall. In fact, maybe what Wynn was talking about is for $75, you're a member, and if you're participating in a group, you have access to that development and you can use it, of course, in your own work as you're exploring, working with it. Once the standard is done, if you were part of the voting group, in this case, D19, you download that copy of it and you got it, and no one's going to stop you from using it. But if you're not a member of either the committee that's voting on it or even working on it, you still have to pay for that standard as well. But that's the beauty. You can actually get that standard with all the other standards once a year if that's the one you choose. So I just want to be clear
Yes,
that.
thank you for clarifying. And that's what I meant to say, is that it's a one time, once a year, and you get everything, not just the one method you're looking for. You'll get access to, everything else too.
You don't. That's my point, is you don't get access to everything else.
Well, you get access to the things that have been published in your section.
In your section,
Yes.
if you vote on it. That's right. You got to grab it when it's on the ballot table.
Well, yes, but if it's been finalized, then it's readily available within your membership realm. There's like a couple different volumes of, there's so many methods that there's different volumes. So when you join, you can join for volume 1 of water, volume 2 of water, and that kind of thing. So you just have to make sure the methods you're interested in are, if they're finalized, which volume they're in, if they're for water, for instance.
And you are required to vote. You can abstain, but
Yes.
if you don't vote, you're going to get kicked out. So you need to spend time engaging in things. And so, yeah.
Yeah. And then Jared shared a video, a link to a video of for the OPC wastewater study participants. Now, I'm not sure, this is something I asked during we had a CASA meeting last week, and this video was referenced, and I asked if ASTM folks, I know there's been a lot of offers made to help on site, both in the field and in the lab. And so I just want to make sure that, if there's problems with whatever method it is, whether it's sampling or lab, that we have, good ability to troubleshoot with the right people in the room or in the field. So, yeah.
Awesome. Thanks for this good discussion, and thank you, Jared, for sharing this video. It's, if anyone hasn't watched it yet, it's quite useful to get an idea of how this method actually works out in the field. It's really, really informative, and I appreciate that.
Yeah, and I think there were a couple other modifications they had to do in terms of the AS team. I think it's a 24-hour collection, and they were able to do it in 12-hour blocks. I don't know if that's in the video itself. And then Bill and Harry Allen from USCPA, who's I think also in the AS team group, they were actively in dialogue with Charles and folks to work through some of these issues. And so I think that's how we arrived at a lot of this. And we tried to do a field study visit, but it didn't work out on timing last summer, but probably we'll aim again for it. But I think things are kind of running smoothly now. So yeah, it'll be interesting to see.
Yeah, thanks, thanks, Jared. Yeah, and the reason we had 24 hours in there for wastewater is to get a whole full diurnal cycle. And again, that was just data quality objective point. And also we wanted to make sure that we could run something for 24 hours without too much manual labor, which in wastewater is really probably not possible. So anyway, thanks, Jared.
Yeah, I think we burned some pumps over the 24-hour cycle.
And some people, right, yeah.
That too.
No. Anyway,
awesome. Well, this is a great discussion, and really thank you, Bill, for presenting. We're running a little bit behind schedule, so I do want to make sure that we be able to get into everything we need to today. Our next speaker, Professor Bob Andrews from the University of Toronto Drinking Water Group, will be presenting on the sampling methods that they've been evaluating for drinking water. So whenever you're ready, Bob, you can go ahead and screen share.
Hey, everyone see okay?
Looks great.
Okay. Yeah, I think what I'm going to talk about now will nicely complement what Bill has been talking about. I'm going to move more into drinking water matrices. We don't work in wastewater matrices, so I think that's going to be one of the primary differences. I don't have to spend much time on this with this audience in terms of, why are we concerned and why are we sampling, other than to highlight in drinking water, a lot of the time we want to go below 20 microns if we can. And in terms of health impacts, I think we need to think about why we are sampling in the first place. We want to determine the physical characteristics associated with microplastics, but we also need to look at the other characteristics, absorption of CECs that may be present, lakes, rivers, also leaching of plastic additives, which in our group we have found that some of the highest toxicity is associated with things that have been added during manufacture. And then also some of our recent work has looked at biofilm growth, including pathogens on different types of polymer surfaces related to microplastics. So again, I think this is more the big picture of why do we sample in the first place? What information is it we want to get out of this? So coming back to, again, reflecting something that Bill was really, trying to state is what's the primary objective when we go about sampling? And we could say, from a drinking water perspective, ultimately it would be human health impacts. We don't want to deliver drinking water that's going to have any acute or chronic health effects associated with it. In wastewater, it may be more directed towards marine life. And again, what we're hearing more and more is just the social and political aspects that people are concerned and aware that there are microplastics in the environment and also in their drinking water. So the questions then come up is, how do we want to monitor it when we're looking at a given system? So for drinking water or wastewater, I think we could say that we'd like to monitor what's coming in the treatment facility, what's going out of the treatment facility, so that we can obtain occurrence data, or we can obtain data regarding treatment performance. And ultimately, if we need to, that will give us insight into which unit processes we may want to optimize to try and remove more microplastics of a given size. Then we go into, should this be a discrete sample or a composite sample? How many times a year should we sample? And these are all big questions that lead into the method that we may use, keeping in mind that it's very expensive and time consuming to analyze the samples. So we just can't go out there and collect 100 samples and analyze them using an auto sampler. So what do we want to quantify? And I think this is particularly important. Are we interested in particle size? For example, what's the minimum size that we can go down to? What's the particle distribution in how many small particles are there versus large particles? How many polymer types do we want to look at? And do we want to do this again by Raman or FTIR, where we get polymer types? Are we looking at total polymer mass using Pyro GCMS? But the limits to that on the low end will be higher than what you see using FTIR or Raman in terms of the number of particles that you can analyze. And when we know the answers, that'll help us really develop what strategy that we should follow. So this is something that I know Scott has mentioned many times before, how there's a lot more smaller particles when we look at particle size distribution, especially those less than 20%, 20 microns. And here's just one study that I pulled that shows, about a third of the particles are less than 20 microns. So should we then be focusing on that size range? Again, it all depends on what the end use is for this data. But it does also infer what type of sampling method to use. Should we be using something that's down in the 2 or 5 micron? Well, that in turn also depends on what sieve size or meshes are commercially available. And from our experience, we can get some in 2 micron, others, the lowest we can go is 5 micron. So that's, some of the data input that we have to think about. I like pulling up a slide like this where data is compared from previous drinking water studies. And again, these just focus on drinking water. I'm looking at just a couple of the more well-referenced studies that have been conducted over the past or reported over the past couple of years. So if we look at this and we look at 3 different studies, for example, you'll find a very, very wide range in terms of the total number of particles in both raw and also treated drinking waters. So then it's interesting to go to an article where they've used the same method, but at multiple treatment facilities, and then just see how that information compares. And in some studies just have said, well, we didn't find anything, it's less than our quantitation limit. But what causes this? Well, if you look at the lower size limit that they were examining, you'll find that the highest concentrations are associated with the smallest sieve sizes, because there are so many more smaller particles when we get down to the lower end. And then we'll see diminishing numbers as the mesh size increases in size. And this is really important because if you look at the data, we're going over 3 to 6 or 7 orders of magnitude when we compare different size lower cutoffs. So I think it's important to always ask the question, what sampling methods were employed? Was this a grab sample, a composite sample? What was the mesh or sieve size that was used? And how much volume of water was passed through it as well? So I think these are really some of the fundamental questions we always have to keep in mind. So if we look at the article by Coleman's, which is really a great one that I reference quite often in saying, where are the knowledge gaps in terms of standardization of sampling? And a lot of them in terms of obtaining low scores were in sampling methods and sample sizes, as opposed to everything else, as well as, for example, the use of spikes or positive controls. So these are the types of things, that we really kept in mind when we started looking at sampling methods a couple of years ago. So he goes on to say that, he suggests minimum 500 liters for surface waters. However, it could quite realistically be that you'd need more than 500 liters, especially if you're looking at less than 1 particle per liter of water. And for tap water, for very clean waters, again, you'd need at least 1,000 liters. Well, that's nice to say, but then how do you filter that amount of water? Which is the minimum size cutoff in terms of sieve or mesh size that you're going to use, and how long is it going to take you to filter that sample? So this is where some of the knowledge gaps come in terms of sampling. I think it's great that ASTM has come up with and done all of the work they have at coming up with a standard practice for sampling. And I appreciate that a lot of it's being related to the wastewater industry as opposed to drinking water. And the 2 methods, of course, include non-pressurized systems where you'd use a pump, and pressurized systems where you would connect to a tap and then pass the water through a stack of sieves. And in terms of looking at the sieve sizes, well, that depends on the type of water. Again, for wastewater or surface water, you may go from 7 sieves down to 5 sieves. And then for drinking water or tap water, go down to a couple of sieves. And then I think the question always is, well, what is the smallest size we should be looking at? And I know from a number of conversations with Scott over the last couple of years, there is some concern of when we get down to the smaller size particles, less than 20 microns, but it makes the whole sampling and analysis protocols all that more challenging. So what we set out to do, and this again was 2.5 years ago now, was to take a look at on-site methods that we could use to filter samples as opposed to bringing samples back to the lab, because we couldn't easily ship like 1,000 or more liters back to a central lab. And compared to what we called inline to in-lab filtration methods. And when I say that, inline, I'm referring to a closed system where you can do the filtration on site, or in lab where you would bring the sample back to the lab. And again, what we wanted to do is maximize the recovery, minimize potential contamination, for example, from airborne sources. And then actually try to validate the use of these closed inline systems, at full-scale facilities. And I'll give you some examples of that. So in terms of in-lab, how we started out was using actually stainless steel kegs. We found that these to be quite durable. We could ship them very easily and they wouldn't get broken. All the surfaces were stainless steel. And here we just show sampling from some pilot scale facilities where we're looking at different filtration combinations. And then we get them back to the lab. We spent a number of months looking at how do we get the microplastics out of the container, looked at different types of surfactants and weighed the pros and cons of that before we directly filtered. And just for example, I'm just using a 10 micron filter, but we've used everything from a 0.2 micron filter and upwards. Or do we pour the water through a stack of sieves? That's something that can be done, in lab or on site. So again, this was kind of our starting point. So what we wanted to do is to see if there was an alternative to this method, which for us, even looking at surface waters, lakes or rivers, took a long time to do. So we said, well, is there another alternative? And us, as well as others, have looked at, using closed systems where you have either, a polycarbonate filter or a stainless steel filter housed within a stainless steel filter system, which is closed. So you can assemble this in a clean space in the lab, take it to the field, do your sampling, then bring it back to the lab, and then open it up there. So this is how we started with things. And because we wanted to increase the surface area such that we can increase the amount of volume filtered, we could look at this in terms of either doing this in series, where we would have a larger filter prior to a smaller filter to avoid clogging, or also do this in parallel just to increase the overall surface area. And we found that we could, for cleaner waters, go anywhere from about 100 to 300 liters, but we still had a desire for larger volumes. And if anyone's interested, this came out in Chemosphere earlier this year. So we did some recoveries where we looked at the couple of methods, used a number of different fragment and fibers of various sizes. And again, what we were doing at that time was spiking them into a keg in a known amount, and then seeing what our recovery was after we had done the filtration. So in comparing the two methods, the in-lab where we'd bring the sample back, or the in-line where we would put the sample through the filter on site, again in a closed system, for a range of particle sizes and polymer types, we found that we got better results using the inline system. So this was, kind of, a good trend that we were on. So we found that we got better recoveries than bringing the sample back to the lab and putting it through a number of open sieves. We averaged over 80% recoveries, lower standard deviation, and greater improvement for various types of polymers. So we also wanted to take a look at contamination, because we always would have field blanks that where the container was open for the same amount of time as it would take to collect a sample. And we had seen that, you know, when we would have to analyze the field blank and then subtract that from what we saw. And again, we compared this, the inline method to the stack sieve method. So in terms of just looking at some of the results, comparing in lab, this would be the stack sieves versus inline, the closed system. One of the things you have to remember is that you need to subtract the blank, and you know, this will account for example, airborne contamination, which we found to be present, especially when we were thinking of fibers. So after we did a blank subtraction, you know, we looked at the results and found that the inline was a better method, again, for the types of waters that we were looking at. So low concentrations, you know, I would say a high concentration for us, maybe 10 microplastics per liter, going down to something like 0.001 per liter. And we found a lot of fibers, but we're not here to talk about that today. So what we did was applied this at a couple of field sites. We tried to determine which treatment technologies would likely have the best removal of microplastics, and that's why we focused on ultrafiltration. And we looked at a couple of facilities where they had different types of pre-treatment, and these were on different source waters prior to ultrafiltration membranes. And here's one of the sites where we use this closed system. And again, others have reported this in the literature, various combinations of, you know, filter types, but we found it to work quite well. And here's just kind of a breakdown between influent and effluent on either side of an ultrafiltration system in a full-scale, quite large water treatment facility. And we looked at, you know, 13 polymer types and found about a 95% removal. And you can also find this published in Chemosphere as well this year. So we still wanted to go higher in terms of the volumes that we could filter. You know, getting over 1,000 liters is our goal. And we wanted to do this when going smaller than 20 micron mesh sizes. So there had been some use of these canister types of filters in the literature. We decided to look at that. Here's just kind of a breakdown of the components. We've assembled these so that we can use them either in parallel or run these in series where we have different mesh sizes. And that's just a picture of, I believe this is a 2 micron size filter. And then here's the canister. And what we do is we have plugs, stainless steel plugs. So these are, again, clean when we ship them out. Then the plugs are put back in and the container is shipped back to the lab so we can open it in a contaminant-free location. And here's just an example of how we have shipped these around the country. And they seem to, you know, survive quite well in Pelican cases. So just a summary of this, the inline closed system appeared to be superior for our applications. We got higher recoveries, lower particle losses, lower atmospheric contamination because it was never open in the field. And it was a simpler procedure that could also decrease analysis time because we could hopefully omit the need for a blank. And again, we could eliminate the need to transport larger samples. So this is kind of my last slide, and I just want to summarize overall comments. Again, the idea was to define an appropriate sampling strategy for a given set of objectives. And these will vary depending on what you're looking for. So, you know, there's no question that this is going to depend on the source water quality and also the level of quantification, whether we use, a stack sieve type, whether we use these smaller scale closed filters, or whether we go to, a larger canister type filter. The volume that we can put through obviously depends on the filter mesh size, as well as the applied flow rate or pressure. So we've done a number of studies where we've monitored pressure on either side and flow rate to try and make sure that these are optimized without actually compromising the integrity of the filter by inadvertently pushing particles through them. Also, we can use parallel filters if we want to increase the volume processed because we have larger surface areas. And we could also use filters in series such that we could reduce the amount of clogging. And whenever possible, what we have found that it's great if we can try and do a trial sampling or initial sampling to give us an idea of how well this is going to work and then adjust some of the parameters. And we've been working on coming up with an automated system that can regulate flow rate, pressure differential across the filter, and we should have something available within the next few months that also gives a tie-in to software that allows us to collect the volume treated remotely. And that's pretty much it. So as Scott said, we're hoping to begin work with DDW comparing this canister method to the stack sieve method, and this is for drinking water facilities. We're also currently doing a survey across Canada where we're comparing performance at drinking water facilities, and we're looking at Raman versus Pyro, and also a number of surrogate parameters like particle counting. We're also looking at microplastics as contaminant vectors. We're looking at the sorption using non-targeted analysis of a couple of hundred organic compounds that may be sorbed from surface waters. We're specifically looking at sorption of PFOA, PFOS, and polyaromatic hydrocarbons, as well looking at leaching of plastic additives. And we think, as I mentioned before, that some of the major toxicity may be associated with these. And for that end, we're doing toxicity assays where we're using different mammalian cell lines. And just a credit to the people that fund all this. And I think that is it. So with that, I would love to take any questions.
Very interesting, Bob. Always great to hear how quickly your work is progressing. I didn't know about the automation that you're developing. That's really exciting. We've got a few questions, but we are running a little bit short on time, so I want to limit this to just a few here. So we've got a question about the atmospheric contamination. So just a little bit more clarification, I guess, on how the closed system eliminates the cross-contamination. And then I guess a follow-up question sort of related is for the recovery comparisons, the inline versus in-lab, are the differences significant or something that can be expected?
Yeah, well, what we're trying to do with the closed system is not have, open air contact, at the facility where we're trying to put, a few hundred or 1,000 liters through a set of stacks, sieves, or conversely, sometimes it may take us an hour to collect a sample, from a pilot scale equipment where we're looking at filtration optimization in terms of granular media filters. And so we would leave the cover off the container for that amount of time. And we found, depending on the site, that there were various quantities of atmospheric borne plastics that we would see in our blanks that we felt we could eliminate by having a closed system that we would take to the facility. Another question in terms of, also, was it significant? Well, we've done spike and recovery test, and it appears, you know, to us that the closed systems, we get better recoveries in both the smaller scale ones, the 47 millimeter diameter, as well as the canister type filters. In terms of pressure, we have seen that the canister type filters will take up to about 70 psi. Beyond that, what we start seeing is deformation in the filter cartridge itself. And that's why we're trying to design a system that makes sure that that doesn't happen. For example, as the filter will clog or blind off, we don't want to inadvertently push plastics through the mesh or collapse the filter cartridge it is as well. In terms of using, you know, surrogates, what we've done is we've done, you know, particle counting from a number of the waters that we've sampled. But when we do particle counts, we quite often get thousands or tens of thousands of particles per liter, where only a few or maybe 10 of those will be microplastics. So we haven't found that bullet that's a good surrogate yet, but it doesn't mean that we've stopped looking.
Awesome, thank you, Bob. And it looks like you're engaging with the chat. So if people have additional questions, feel free to respond to those in the chat. We'll move on to our next agenda item here. So we're going to have some quick updates from our work group leads. First is Brent Alspach will be walking us through what the consumer messaging work group has been doing. And Brent, go ahead, free to unmute and share your screen. You need to.
Thanks, Scott. Can you hear me?
Loud and clear.
Okay, so hopefully everybody can see my screen here, and I will keep this relatively brief. Acknowledge.
We can't see your screen.
Let's see. How about now?
Looks good.
Thank you. So I will keep this relatively brief, keeping in mind that by far the most interesting part of this call was the speakers that preceded us. So I'm happy to have ceded time to their purposes. And what I really want to do with the time I have today is just to update the broader group on where the work group is going, and also potentially use this as a recruiting tool for anybody else who hasn't already joined the work group to do so if you feel like it's something to which you could contribute. So with that in mind, I think I showed these slides in the previous time we had a big group meeting, but this just indicates the content of what the entirety of what we're going to do. Scott, I think initially you identified Terry as one of my co-leads, and also my colleague Alma, who is likewise working with me on the Water Research Foundation messaging project as well. We also want to identify the audience, of course, for our work, what the goals are, the end work product, which is to say the deliverables, the platforms by which this work product would be stored, and how we disseminate the word that this resource generated exists, how we do it, or the approach, the timing, and logistics. So I'm gonna get to this. So in terms of a goal, following the last meeting, I believe, which was in March, we talked a little bit on my co-leads and Scott and Monica, and determined that probably the best thing for this group to do to serve the community was to develop language for utilities to use in their consumer confidence reports, particularly relevant for California, where utilities will be sampling, and potentially other states around the country, either by decree or if they choose to do their own sampling and then disclose results in their CCRs for whatever purpose they may feel is appropriate. But not only that language for the CCR, but also some supplemental context about microplastics that might be useful for utility reference. And that way they have a little more background knowledge, or they have additional information that they could supplement the CCR content that we would provide if they chose to use that. And then we developed on our previous leadership call some brainstorming points, and just to run through some of these things in the context of developing this CCR content. And we're not going to answer these questions today, of course. This is just to give the broader group an understanding of where we're going and the kinds of questions that we have to address. So how much content should we develop for this resource? What types of information should be included or are appropriate for inclusion? I don't mean that with respect to microplastics specifically, but what categories of information, which is to say toxicology for one, should that be included or avoided? For the next bullet point, what types of information should be explicitly avoided and or are not appropriate for inclusion? And then what are the intended messages we're trying to convey to consumers in their CCRs regarding microplastics? And that's probably one of the most seminal points we'll need to address. And then what behavioral changes, if any, are we hoping to promote through CCR messaging that we have an opportunity to affect? And continuing some of the points, how can we leverage this unique scenario that we have to increase the public trust in drinking water, knowing that in this case, we have this emerging contaminant, which consumer perhaps concern will only increase over time as more results are disseminated. So how can we leverage this again to make the consumers trust drinking water more than they currently do? What general CCR language will be generated by the state for guidance, or this sort of a stock language, if you will, and I'll get to that in a subsequent slide. Should the product include background or context as an FYI utilities? I think that we've decided that it should. The craft own messages or provide specific language for inclusion, or both. In essence, should we just say, here are the types of things you should include and you can formulate the language on your own? Should we just provide specific language to say you can cut and paste it, or potentially give them a couple of options when utilities could either cut and paste or craft their own messaging per our guidance? And would additional research on risk communication of microplastics is useful or relevant? And what strategies have proven useful for risk communication? And Scott's been very helpful from some of his interactions with Dr. Sabine Paul on that point as well. And after the last call, we know the state has generated that language, which Scott did provide. And as it stands today, I'll just read this quote. Studies of rodents exposed to some types of microplastics through drinking water indicate potentially adverse effects, including on the reproductive system. However, more research is needed to understand potential human health implications and at what concentrations adverse effects may occur. Therefore, California is monitoring microplastics in drinking water to understand its occurrence and supporting ongoing research. So essentially, this is what the state will provide utilities for their use if they want it. And the goal for our work group is to supplement that potentially with more information, either verbatim or conceptual or guidance for utilities to include if they choose to do so. So next steps, recruiting, which is really what this is now, probably a kickoff call subsequent to this in July, once anyone else who wants to join the group has indicated as such, and then execute the work in the summer and fall, and then potentially beyond that as circumstances. And with that, Scott, if there any time that we have, I'm happy to entertain some questions either here or offline.
Thanks so much, Brent. If anyone has any questions, feel free to answer or ask in the chat, or you can also just unmute and scroll away. Got a couple minutes.
I see some folks indicating in the work group that they would like to join. Either Scott or Monica, are you keeping track of those things for the database?
I haven't seen that in the chat. Was it a direct message?
Direct to me, I'm sorry. I'll try to keep track of those. And then if anybody else indicates, just if you can capture that and I'll update our spreadsheet, that would be terrific.
Alrighty, and if anybody has any questions, feel free to listen to the chat, reach out to Brent. One last chance. Otherwise, we're gonna hand it over to our next speaker, Shelly Walther, will be giving an update on our working group.
Thanks, Scott. And I'm going to give you an abbreviated version just so we can keep to our agenda, our time. And so I'm going ahead and share my screen. All right, can everyone see my slides?
Looks great.
Okay, great. So just a brief update on where we're going and what we're thinking about the playbook. Just as a brief recap, it will be a living document. We do have definite priorities for year one, which we put out another version of the survey during our last meeting, and we got some great feedback. There were a lot of drinking water participants in that meeting, and that was the overwhelming message that drinking water and also sample design were the major focus that people would like to contribute to, and they felt the most need was there, just in terms of future and current regulatory requirements. And so, the tenets of the playbook would be that we would be building on current best available science and lessons learned, and that it would be question-driven, evidence-based, and statistically rigorous information to lead you to those types of studies. And so the universal parts that transcend any matrix, whether it's drinking water or biosolids, would be, overall study design recommendations, sample collection, reporting, sample analysis, and then data reporting and analysis and reproducibility. And this is a straw man and an outline of the playbook. It's a little bit more detailed than what you've seen before, but the pictures on the right kind of illustrate some of the things I'm going to touch on today that are considerations that would go into the playbook. So things, it will give a really good sense of how to design your study depending on what the questions are. And it will go into the methods that the State Water Board will be finalizing, as well as ASTM and other methods that are validated and have been put out there, and also some pros and cons, those kinds of things. So it will delve into data capture and standardization. If you're reporting it, if you're required to report your data, how would you do that? It'll have sample templates in it, for instance. And so it's meant to be a very useful guide. And so just touching on the sample design, making sure that any part of making sure that data are reproducible starts with your sample design, as Bill Robberson was saying earlier. And so when you're looking at some of these things, you're thinking about designing your study. You have to think about, well, what is the end use of the study, and what are the questions being asked? Now, these may be questions that are set forth by regulators. They may be set forth by the public that want to know. And there are a lot of different end uses. Is it a one-time study? Or is it every 5 years or every quarter? That'll make a difference, let's say, in your sample collection. If it's a very laborious method, maybe you don't want to do that quarterly. Maybe that will, and the sample analysis, if it takes a week per slide, you may not be able to do that for long-term monitoring. But if it's a one-time study and you have a certain budget in mind, that'll all come into play. But of course, all of these things need to be driven by reproducibility and sample representation, including in that. So just an example of field studies, one recommendation is that a new microplastics monitoring program could very well be incorporated into existing monitoring programs. And so that it's integrated in terms of the field sampling and regional surveys. So maybe if you're out collecting one type of sample, you're also collecting another type of sample with contamination in mind, and robust methods. And then in terms of uncertainty, this is something that has been talked a lot in other fields. And in microplastics, we know that there are a lot of sources of uncertainty, such as contamination, and limited sample matrix. So if it's really expensive to analyze a sample, then maybe there won't be that much sample that can be analyzed. And does that affect the end result? This, hopefully this will help answer some of those questions as a go-to, source for that kind of information. And this is an example of that. When you're measuring uncertainty, you're looking at the concentration and of microplastics. It'll vary depending on how much microplastics are in the sample and how much sample there is. And there's a way to measure some of these sampling biases. The other thing is looking at precision, and precision, of course, is being able to hit the same target, multiple times. And if you have one sample and it's analyzed by one analysis, let's say a scan of an FTIR, and then it's analyzed 6 more times, you would expect to get the same result. But that actually, what the California Department of Health and US EPA Region 9 are seeing is that's not necessarily the case. And they're coming out with a paper looking at precision estimates. And it could have a profound effect on how we measure microplastics, and especially with IR, but also with mass. So in terms of counts or mass, it's important to think about precision and how it's measured. Using relative standard deviation is one way of looking at it, and just to ensure repeatability and have some knowledge. If you've run it once, do you know for sure that that's the answer? If you've run it 6 times and that's a known thing, okay, maybe you don't need to run it 6 times next time, but having that kind of information for your instrument and your instrument type is pretty critical. And but also the interpretation of an RSD, or relative standard deviation, needs to be kind of better explained, and kind of maybe we can take Bill and Harry up on their offer to come and speak with the subgroup about some of these issues and how to tackle them. And luckily, this is not, even though microplastics it's been challenging to kind of center on methods and things, there have been existing particle count standards, such as with this asbestos. So we do have, that's through the US EPA. So there are some ways of looking at how other people have standardized this, and maybe they don't have the same challenges as microplastics, but it's a step in the right direction in terms of data analysis. And so that's something that we could outline in the playbook, is looking at different ways of doing precision estimates for both counts and for mass. And touching more on statistical rigor of these studies is making sure that we're not encountering any zombies of data analysis. And I took these from the 40 years of water quality statistics, a presentation that Dennis Helsel gave, and there's a link below. You can, when this presentation is stored in the Google Drive, so you can find it there. But he has a whole book available of some of these things, just things to be aware of that we can incorporate into our study design and also our analyses. And so all of those considerations will be made, and they will guide what's cut off at the bottom intentionally. What methods are we talking about? It depends on the management question and the monitoring question and everything else. And I took this from the trash monitoring playbook. It's all relevant to microplastics as well. So our next steps, I'm still looking for a co-chair who's willing to, be on the the headline organization, helping with that kind of thing, organization admin, and maybe you have a focus on drinking water, because that's going to be kind of the first matrix that we really focus on. We're going to be rolling out the Zotero literature database shortly. And we're looking at a July-August timeframe for, I call it the MPMP, the Microplastics Monitoring Playbook. It's, it seems it's easy to type, so I've been using that. And, we can focus on drinking water matrices and talk, delve a little bit deeper into this data reproducibility and some key recommendations that we could, from lessons learned in the past. And that's all I have for right now. So questions?
Really exciting, Shelly. You put a lot of thought into this. Really appreciate that. We do have a little bit of time for questions. Again, people can unmute. There's a couple in the chat. One is on precision. Have you considered to include accuracy? One can be precise but not accurate.
So when you have an unknown, it's hard to measure accuracy except for using spiking the sample. So with spikes, you can get a good accuracy measure, but with unknowns, precision, doing repeated samples is one way of really getting a good estimate of your sampling error.
We've got another question from Terry. Will the study be completed in enough time to use as a framework to develop the monitoring plans for specific drinking water systems to develop monitoring plans? Will it include a monitoring cost analysis that includes labor costs? It'd be helpful for project and monitoring plan budgets.
Yeah, so by design, we need to meet the needs of the State Water Board, and we have kind of a one-year timeline. It's next spring. So that's when this is going to be, there's going to be a lot of discussions about it and meetings and drafts, that kind of thing. But we want to have the, something ready to use for that audience, for the drinking water community in particular. We know that that's coming up. And so, yes, that's, Scott has already, asked that we have that ready to go by next spring. And so that's the goal, to have, to speak to that need in particular. It's a great question.
Alrighty, and I think we are running a little bit short on time right now. I do want to get to the last item that we have, which is Dr. Wynn Cowger is going to give us an update on the data harmonization platform really quick. And then hopefully we'll have a little bit of time for general discussion if there's still any questions left. Again, folks, you can write questions in the chat if it didn't get answered before.
Thanks. And yeah, Scott, I shared the link in the chat so we could share that with everybody. And I'll try to go real quick. So thanks a lot for the invite. I'm going to be talking about an open source data portal that we're developing for microplastics in drinking water, and specifically focused on California right now. And I'd like to acknowledge the collaborators and funding. We have our funders who are also active members of our group, from California 100 and the People Lab with us. And that's really awesome. So thanks for being here. I'll let them speak up too at the end if they want to add anything. So like we've been talking about, California is going to be accrediting a method for testing microplastics in drinking water. They need to share data for that method, and that data should be open source or open access. But microplastic data is really challenging to analyze. We have a lot of different metrics that we like to look at for microplastics data. So you can't really do this kind of analytics or store this kind of data in a really standard data format. And state policies are now requiring software to be developed open source. And so some of the examples here, I've been reading up on this quite a bit now. So section 5160 requires that open data be shared by all California agencies. And then the newer one is requiring that software be open source by default by all California agencies. So this is a really exciting development. We're excited to be a part of it. So in a nutshell, this data portal will allow accredited labs to share data to a CCAN data portal. It's going to be developed open source. And we're also going to add in many of the data analysis tools that some of you probably are using that we've developed in the past: OpenSpeccy, Tomex, Plastiverse, Trash Taxonomy, all these other softwares. We're going to be adding in analytics from those. And then the data portal is going to spit out data in a format that can go to the other state data portals like CDIN and SCWIS. And this is basically our timeline here. So we are currently, this is part of the stakeholder input we're gathering from you all. So please feel free to email us afterward if you have any input that you can't get to. We're going to be developing a minimum viable product after this, and then do about a half year of beta testing with folks. And then we will finalize and publish it. And so that's all part of the innovation part of this project. We're also doing research as part of this. So we're going to be scoping the research for the next 6 months and collecting data throughout the entire project. And then there will be a couple of publications that'll be submitted. And so as part of the stakeholder input, we're also doing workshops in addition to just giving presentations like this. And so we have our first workshop coming up for people who are interested in helping us to develop the open source platform. It is going to be, we're going to be piggybacking on the California Water Data Challenge, which does have prizes associated with it. I'm not sure if they're monetary, but they're definitely like, you know, you get a props somewhere on the web. And June 28th and 29th, we'll be hosting like some sessions during the California Water Data Challenge, where you can come and ask us questions about the data portal and try to develop your own project and team around something to help us develop out the data portal. So next steps, I'll drop this in the chat too. You can get in touch with me, Win at moreplasticresearch.org, and then I'll drop the link in the chat for signing up for the Water Data Challenge if you're interested in that. And we'll be checking back in future work group meetings to let you know about our progress over the next 2 years. So thanks a lot. And Rohit, Ryan, I don't know if you wanted to add anything. Oh, good. Okay. Sweet. So if anybody has any questions, can take those. Let's see. Oh, thanks, Scott, for dropping that in the chat. Cool. I don't see any hands raised or any questions, so I'm sure everybody's just frantically signing up for the California Water Data Challenge right now, so that's awesome. All right. Great. Thanks. Feel free to get in touch.
Yeah, thank you so much, Wynn. Yeah, folks, if you haven't been to one of the data challenges before, they're really exciting. This year is focused on diversity, equity, and inclusion, and how we can meet the demands of the most vulnerable communities. So it's going to be a really exciting meeting. It's a three-day symposium at the end of this month. I mentioned before that we have this folder, Google Doc folder, that has all the information from today's meetings. Feel free to check that out. It'll have the presentations, slides, recordings, some notes, as well as some additional publications relevant to today's meeting. So I dropped that in a link. A few other links that I dropped in here. People might be wondering what those are. Plastiverse has been mentioned briefly a few times today. This is a website that we just launched. It's a collaboration between Wynn, myself, Leah Thornton-Hampton, who's also here, Kara Wigan, Restus Herodotu, and then Cleo Stratman. This is a website that is designed to act as a centralized living repository or hub for plastic pollution research. It's just getting started right now. It really is just an index of all of the websites that are out there that we're aware of, and making them accessible to folks. But this is a community effort. that we hope to build in collaboration with you all, as well as the greater microplastics community worldwide. And I see Leah just popped on. I don't know if she wanted to say anything about that. We also just released TOMAX, the Toxicity of Microplastics Explorer app, that went online, I think last week. So be sure to check that out. Also drop the link in the chat. The publication for that has also just been released. So if you're interested in reading that, it's online. So we have a few more minutes. If anyone wants to speak up, this is, more for open discussion. Yeah, I'll hand it over.
I'm interested in asking the group if anybody here is does like software development on the side and would be interested in collaborating on developing any of these projects within our ecosystem. We're constantly looking for more people to join the team. Since we have so many people here, maybe there's a chance there could be somebody that we haven't gotten in touch with yet.
Hey, one thing I'm seeing in the chat is it seems like the state of California browser is not allowing the Plastiverse website to open. Can we maybe talk to someone in IT over there to approve that website or something?
Yes, that is quite interesting. It's a WordPress website, so it's interesting that our IT department is banning it. I don't know if anyone else is having trouble with it.
Yeah, so we'll note that and try to find, I guess, who can deal with it.
Yeah, I think that's going to be on the state of California, really. I don't know, Wayne, if there's something that we can do to have it not get flagged as a fishy website.
Yeah, do not know. I mean, we could check the like robots document to see if there's some sort of weird thing in there. But I don't know. I can take a look and see if I can find that.
Awesome, thanks, Melissa. Rick just asked a question: will the sampling pilot BC Brown Caldwell is managing include closed system sampling and or OpenSIV? So if Bob Andrews is still on the line, you might want to go ahead and answer this one.
Well, that's going to be looking at comparing both OpenSIV and closed.
I don't know, Rick, did you have any follow-up on that, or just a curiosity?
No, Scott, I mean, the closed system's obviously very interesting, eliminating the air deposition. So that's great that you guys are looking at both. That's going to be a great data set.
Thanks, Rick. And just a reminder to everyone, these large group meetings that we're having today, these are occurring on a quarterly basis. So soon I'll send out an update about when our next meeting is going to be. It'll likely be at a very similar time, 11 to 1. Seems to work well for most people. The individual work groups have different schedules. So if you're interested in being involved with those, make sure you reach out to the work group leads and figure out when those upcoming meetings are.
And we'll be sure to broadcast the meeting dates through the listserv, that the same one that's used for the microplastics monitoring work group, so that we're all on the same page.
Thanks for dropping your contact info in there, Leah. Alrighty, so that's effectively all we've got for today. Terry asked when the next work group meeting is going to be. We haven't finalized the date on that, but it'll be approximately 3 months from now. I'll send out a message probably within the next week for that date and make sure we get on everyone's calendars. Again, that's all we have. Thank you all so much for attending. Thanks to our speakers, work group leads, and everyone that's making this collaboration happen. This is a really exciting effort that we're building here.
Thanks, everyone. Great input, great questions.